Welcome friends to this second lecture of week 12 of this NPTEL online certification course of environmental soil chemistry, and in this week we are talking about analytical techniques for assessing soil pollution. So, in our previous lecture we have covered the the following concepts. We have started with the choice of techniques for soil analysis and why soil analysis is important, and then we talked about the sample preparation, and then we started with the determination of inorganic pollutants. So, we started with the nitrogen, and we will try to finish the nitrogen today, and then we will start from phosphorus and salts and so on so forth. So, in the previous lecture, the first lecture of this module 12, we have covered these basic aspects of soil analysis, and then what are the major choices of soil analysis technique, what are the major considerations like sensitivity, specificity, accuracy of calibration, cost effectiveness, specific purpose of the parameters to be determined in the analysis. So, we have discussed all these things in details. And then we have talked about some basic instruments like spectrophotometer, flame atomic absorption spectrometry, flame atomic emission spectrometry, ICP-AES, then HPLC, and also HPLC-MS like mass spectrometer for different pollutants, for measurement of different pollutants. We also talked about the auto sampler, which is required for automated measurements without human interventions. So, this auto sampler is an integral part of different high valued instruments, which generally helps for automatic sampling, automatic sampling for subsequent analysis. Then we talked about the sample preparation, why it is important, and we have covered different steps of sample preparation like sample description, drying and complete samples, drying of the complete samples in the air, oven drying and freeze drying and all these things. So, we have discussed the freeze drying method, why it is required for certain elements like arsenic, like mercury, like selenium, and some other organometallic compounds which are easily lost upon by air drying. So, that is for these organometallic compounds and these elements we generally go for freeze drying for sample preparation and keeping the samples for a long period of time. Then we have talked about the sample crushing, sample homogenization, milling process, crushing process, why it is required, for which condition we will go with the crushing, for which when the test sample is less than 2 gram we will go for the milling process, and the size should be less than 250 microns for this case. After the milling, we have talked about the sieving and why it is required for the sieve, why it is preferable to have plastic sieve instead of metallic sieve, we have also discussed. Then we have talked about the subsampling and storage, what is the quartering method I have discussed, and then what are the consideration for keeping the samples for a long period of time, and what is storage we have also covered. Then we started with the determination of inorganic pollutants like nitrogens, and we have talked about different extractants like saturated calcium sulphate to H2, then calcium chloride, sodium bicarbonate, copper sulphate, and so on and so forth. And then we have talked about some analytical methods or instruments or tools which we require for inorganic measurement of inorganic nitrogen. So, we started with the ion selective electrodes. These ion selective electrodes are basically membrane based electrodes that are specific, that are sensitive to a specific ion in aqueous solution. In this case, this is a nitrate ion. So, when the membrane of the electrode is in contact with the solution containing the specific ion, a voltage difference or voltage gradient is developed across the membrane, and that is basically measured in these electrodes, in this ion specific electrode or ISE. Then one of the major drawback of this ion selective electrode that sometimes interferences are there. So, frequent calibration is required for this type of electrodes. So, today we are going to start with this new instruments. I mean, this is another instruments which we use for analytical, for measurement of inorganic nitrogen. We call it continuous flow analyzer, or in short form CFA. Now, this continuous flow analyzer basically operates on the basis of continuous flow analysis. Now, this continuous flow analysis or continuous flow analyzer operate by introducing a sample and reagents into tubing separated by bubbles. I will show you one picture. Now, each segment of sample mixture goes through a mixing coil or other tubing where chemical reactions basically occur. So, various modules can be introduced to perform specific chemical reactions, and the sample is then transported to a detector where the developed color. So, there is a color development, and this developed color is basically measured, this developed color is measured and then sample is analyzed in a flow cell. So, this is basically the principle of this continuous flow analyzer. Again, in this continuous flow analyzer, the segment of sample mixture goes through a mixing coil and other tubing where a chemical reagents basically mix with them. So, these samples are separated by bubbles. This is how this continuous flow analyzer basically operates. You can see here, this is a continuous flow analyzer and this is a auto sampler for this continuous flow analyzer. Now, as I told you that the principle of continuous flow analyzer says that through this tubing the sample and air bubbles are basically circulated. So, as you can see, samples are separated by these air bubbles. So, the samples are moving through these tubings, and the reagent is also pumped in this tubing in the certain points of the tubing. So, here in this point there is a mixing between the sample and the reagent, and obviously after the mixing there are some chemical reactions, and after this chemical reaction they move to the incubator and reaction detector where they are being detected by the development of certain colors. So, this is how this continuous flow analyzer basically operates. It is basically the operating principle of this continuous flow analyzer. Now, there are several techniques of continuous flow analyzer such as segmented flow method or segmented flow analysis, in short we call it SFA, which basically use turbulent flow conditions that allow for complete sample dispersion. So, this SFA or segmented flow analysis basically deals with the turbulent flow condition that allows for complete sample dispersion. And other technique for this continuous flow analysis is known as the flow injection analysis. In short form we call it FIA. Now, this flow injection analysis and sequential injection analysis uses laminar flow existing in the narrow bore tube to mix the reagent to eliminate the need of air bubble partitioning. So, there is no need for air bubble partitioning. So, there is a laminar flow existing in the narrow bore tubing basically to mix the reagent, and there is no need for air bubble partitioning. So, this is called continuous flow analysis, and also the instrument is known as this is called the flow injection analysis, and the instrument is known as flow injection analyzer. So, this is one type of continuous flow analysis. Okay. So, another instrument is there for measurement of inorganic nitrogen, that is called discrete analyzer. Now, what is this discrete analyzer? Now, it is contrast to this continuous flow analyzer. This discrete analyzer basically keeps samples separate throughout the testing process. So, in the continuous flow analyzer, you are seeing that the instrument basically flows the samples continuously. However, in case of discrete analyzer, they separate the samples, they keep the samples separated throughout the testing procedure and dispensing precise amount when is required. Now, rotating individual cuvettes through the instrument instead of releasing the sample in a continuous stream. So, basically when they basically rotate the individual cuvettes through the instrument instead of releasing the sample in a continuous stream, that cuts down the reagent waste and can produce hundreds of results per hour. So, this instrument is also very precise, and it requires a specific amount of specific chemical, and without going for the continuous flow, it basically takes the samples, it basically keeps the samples separate. So, this is called discrete analyzer, and as you can see here, this is a picture of a discrete analyzer. Now, other method for measurement of inorganic nitrogen. Now, we are talking about the total inorganic nitrogen. Now, in the total inorganic nitrogen, generally we follow two methods. One is the weight oxidation methods, another is Dumas method. Now, let us consider first, let us first discuss the weight oxidation methods or Zeldal method. We know it is also known as the Zeldal method. So, this weight oxidation method or Zeldal method is a widely used technology, and it involves the conversion of organic and inorganic nitrogen into ammonium ion by digestion of the samples and the subsequent measurement of the resulting ion. So, in most of the soil chemistry laboratory, you will see that this instrument is present, and this Zeldal apparatus is sometime it is automated, sometime it is manual. So, this Zeldal analysis or Zeldal analyzer involves the conversion of organic and inorganic nitrogen into ammonium ion. So, they basically converts all the organic form of nitrogen which is present in the proteinaceous material and also inorganic nitrogen into ammonium ion by digestion of the samples and then subsequent measurement of the resulting ions. And for digestion, it has also auxiliary heating mantles or digestion block. As you can see in this picture, this is the distillation unit of the Zeldal apparatus, and this is the digestion unit where block digesters. It is basically block digestion process. So, there are the digestion of the samples are occurring in this block digestion chamber. So, this is how this weight oxidation is weight oxidation by Zeldal method is performed. Now, the second method is known as the dry combustion, and this method is based on Dumas method. Now, the instrument which we generally use for this dry combustion is known as CHNS analyzer or elemental analyzers. Now, the CHNS stands for carbon, hydrogen, nitrogen and sulfur elemental analyzers. Now, this is a very sophisticated and accurate instrument. This CHNS analyzer provides a means for rapid determination of carbon, hydrogen, nitrogen and sulfur in organic matrices and other types of materials. So, basically it gives you the total concentration or total content of carbon, hydrogen, nitrogen and sulfur in organic matrices and other types of materials. Now, they are also capable of handling a wide variety of sample types including solids, liquids and volatiles and viscous samples in the field of pharmaceuticals, polymers, chemicals, environment, food and energy. So, this instrument has a widespread use in several industries, not only in agricultural environment discipline, but also in polymers, chemicals, pharmaceuticals, food and energy and so on and so forth. So, these instruments basically has several parts. As you can see here, it has auto sampler, then it has sample inlet system, and there where they basically introduce helium and oxygen gas, and there is a combustion chamber where there is a flash dynamic combustion occurs at temperature of 1800 degree centigrade, where they oxidize, and after they oxidize these nitrogen, carbon, hydrogen and sulfur, they basically converts into carbon dioxide and then water and water vapor and also the sulfur dioxide, and then this mixture of gases are circulated through a GC column or gas chromatography column. And this gas chromatography, when they are moved, when this gas mixture moves through this gas chromatography column, they separate out, and they are, they separate out and being detected by this detector at the end of the GC column to measure the concentration of the total carbon, total hydrogen, total nitrogen, total sulfur. So, this is how this instrument basically works. So, the next slide will give you more information about this CHNS analyzer. The CHNS analyzer is a very important instrument and it is very accurate. So, if you are interested to measure the total carbon and total nitrogen in the soil, you need this instrument, and this very sophisticated instrument too. Now, this instrument, as I told you, is highly precise, accurate, fast and easy operation, and shows the easy operation. And this built-in chromatographic column converts the compound and eluids it in the form of nitrogen dioxide, carbon dioxide, sulfur dioxide and water vapor, which are then detected with the help of thermal conductivity detector. So, this detector which is present is thermal conductivity detector. So, in this diagram it is more clear. So, there is sample holder or auto sampler, and after that there is an ignition chamber, and this ignition chamber, in this ignition chamber these samples are being ignited with the help of oxygen which are being introduced, and then they convert into this sulfur dioxide, carbon dioxide, nitrogen dioxide and water vapor, and then they move through a pre-pack column. After they move this gas mixture through this pre-pack column, they further move through this GC column or gas chromatography column, and finally, they are being detected by this TCD detector. So, this TCD detector is a short form of thermal conductivity detector, and based on the individual compound, this spectrum will be obtained, and from this analyzing this spectrum, they can calculate the total carbon percentage, total hydrogen percentage, total nitrogen percentage and total sulfur percentage also, and sometimes oxygen percentage also. So, this is how this CHNS analyzer basically operates. Now, this instrument basically is calibrated with the analysis of standard compounds using the K factor calculations. Now, thus the instrument ensures maximum reliability of the results, because the combustion gases are not split or diluted, but directly carried to the built-in GC system. So, they are not diverted anywhere, they directly carried to the GC system for simultaneous detection of the CHNS in the less than 10 minutes. So, it is very fast also. Now, this method finds greatest utility in finding out the percentage of carbon, hydrogen, nitrogen, sulfur and oxygen in organic compounds which are generally combustible at 1800 degree centigrade. So, guys, the key components of CHNS analyzers are auto sampler, combustion reactors, chromatographic columns and TCD detectors. So, we can see here first obviously the auto sampler, as you can see here. So, the auto samplers is here, and then combustion reactors, and then chromatographic columns, and then TCD detector. These are the 4 major parts of this CHNS analyzer. So, guys, I hope that you have got an basic understanding of the principle of CHNS analyzer. So, we have started with the ion specific electrode for determination of inorganic nitrogen, and then we talked about the continuous flow analyzer, and also we talked about the discrete analyzer for measurement of inorganic nitrogens, and then for measurement of total nitrogen we talked about the Zeldal methods, which is known for the weight digestion, and also for dry combustion we have used the Dumas method, and this method is basically performed with this CHNS analyzer instrument. So, these all instruments are required when you are going to measure the concentration of different forms of nitrogen in different environmental samples, and thus these instruments are are integral part for any sophisticated environmental soil chemistry lab. You will see them very often, and among these all these four different instruments which we have talked about, the CHNS analyzer and also this continuous flow analyzer and discrete analyzer are somewhat very somewhat costly. However, this ion selective electrode is comparatively cheaper, and also this Geldal apparatus is also comparatively cheaper than this CHNS analyzer. So, if there is a budgetary constraint, people generally go with the Geldal method instead of CHNS analysis, because CHNS analysis is costly. Although it is accurate, although it is precise and requires less amount of time, however, it is costly, and also the consumables, specifically the gases, are very costly in nature. So, this is the a picture of the CHNS analyzer, and so let us move ahead and see the determination of inorganic phosphorus. So, we have covered the nitrogen. Now, let us see how we can measure the inorganic forms of phosphorus in the soil. Now, you know that inorganic phosphorus in soil occurs predominantly as phosphates, and these phosphates basically encompasses orthophosphate and condensed phosphates. And the extractable phosphates in soils is usually recovered by using a specific extractant, we call it Mehlich III extractant, and the procedure is known as the Mehlich III procedure. And this Mehlich III extractant is basically a multi-element extractant, and it is basically used for extracting the soil solution. Now, this Mehlich III soil extractant is basically composed of these components. So, it has 0.2 molar glacial acetic acid, it has 0.25 molar of ammonium nitrate, it consists of 0.015 molar of ammonium fluoride, it also has 0.013 molar of nitric acid and 0.001 molar of EDTA or ethylene diamine tetraacetic acid, which is a chelating agent. So, these all comprises in the Mehlich III extractant. So, this Mehlich III extractant contains all these, and remember the reason for containing all these is all these components, individual component has their specific activity, and that is why the Mehlich III extractant is known for extracting not only phosphorus, but a wide range of other elements also from the soil. And each of these component is known to extract more than one elements. So, apart from nitrogen, these all other important trace elements as well as the primary elements can be extracted by this Mehlich III extractant. Now, the amount of Mehlich III extractable phosphorus. So, let us again come back to phosphorus. So, the amount of Mehlich III extractable phosphorus is highly correlated with the phosphorus uptake by plants in a wide range of local soils. So, the Mehlich III extractable phosphates basically encompasses the plant available phosphorus. So, for soil fertility based measurements also, people generally go for this Mehlich 3 extraction. So, these are the this is the Mehlich 3 extraction method. So, let us see some of the important what are the important instruments for measurement of this inorganic phosphorus. So, the most important instrument for measurement of this inorganic phosphorus is ICP-AES. So, after you extract the solution through Mehlich 3 extractant, this extracted solution is injected into ICP-AES to measure individual ions. Now, this ICP-AES is a short form of inductively coupled plasma atomic emission spectrometry or ICP-AES. Now, this is basically an emission spectrophotometric technique and it is highly precise and accurate. So, this is basically the schematic of different components of the ICP-AES. So, it basically here the argon gas is supplied for generation of the plasma, and why plasma is required we will discuss. And also there is a nebulizer to help injecting the samples into the plasma torch. So, there is a plasma torch, as you can see here. This plasma torch generates the inductively coupled plasma, which is the source of energy, and as a result of that the sample get atomized, and further they are being detected the wavelength which is generated from these excited atoms are basically analyzed by monochromator, then detector and computer, further analyzed by a computer. So, there is a sample solution, as you can see we are injecting the sample solution here, and there is a nebulizer which helps in the introducing these sample into the plasma torch, and in this plasma torch to generate the plasma the argon is being supplied, and as a result of that there is a plasma formation, and this plasma is very high temperature, and this high temperature of the plasma helps the samples to excite the atoms of these samples to excite and emit the characteristic wavelengths of light, which are being subsequently analyzed by monochromator and detector. So, this is the basic construction of a ICP-AES spectrometer. Let us wrap up here, and in the next lecture we will discuss what is the specific function of what is the principle of this ICP-AES and how does this plasma torch basically works, why we call it inductively coupled plasma. So, in details we will be discussing the working principle as well as the construction of this ICP-AES spectrometer in our next lecture. So, guys, I hope that you have learned something new in this lecture. We have discussed some important instruments for nitrogen, and we have started discussing about the ICP-AES. Please stay tuned and let us meet in our next lecture to discuss more details about ICP-AES and why it is called a superior instrument. What are the benefits of using the ICP-AES? After we complete ICP-AES, we will also discuss some other sophisticated instruments too. We will be also discussing the spectrophotometry. Thank you very much. Let us meet in our third lecture of week 12.